Y'all tell me how I knew that it would be like 3 people, even though I know nobody's doing anything at this time because we would normally be in class.
Exactly. Well, I usually study the day after the quiz, so I didn't study that much. So if I get any of these answers wrong on Kahoot, just know I haven't went over these
You're okay. Like I figured a
shit.
lot of
So
people, yeah, I figured a lot of people haven't actually started the studying part yet. That's okay. But I feel like at least this is like recalling information and getting us in the mood for learning, relearning that stuff from past exams. But
okay, good. So I might get a lot of answers wrong, but I'll get them right on the test.
I'm not going to judge yet. Put on this nice relaxing video for my dogs so that they would stay calm and stay in this room. And they've decided that this is the last place they want to be. Just out there barking and having, living their best lives.
Okay. This is not working. Hello.
I mean, there's no drill. I'm gonna wait till probably 5 after for more people to creep in and then we'll get started. And I'm sure people are gonna trickle in as we get going. The wildlife expert um, got back to us yesterday and said that, given the size of the little possum, that he was big enough, he's probably weaned from the mom and is big enough to be released. And he was eating like food, like he told to give him dog food, and so if he's eating food and drinking water on his own, he's good to be released. So we took him, we live like right by Overholser. We can't see the lake, but it's like the street over. So we just drove down to like the dam area over by Overholser, and there's this big like woodsy area on the south side of the dam. And so we took him back there and let him go. And he like got out of a little box and like looked at us and was like hissing at us and was just like backing into the forest. Like,
now he's all badass.
yeah. It was hilarious, but yeah, I think he'll be okay. He seemed like he was doing pretty good. My husband was like, I would keep him if I knew how to care for him. And I was like, I know you would. Because to him, the possum is a spirit animal. So we get so much wildlife here, it's nuts. Even while we were walking like the possum down to the lake, we saw deer. They're just all over the place over here. It's hard if you live over here, then like at nighttime you don't drive down Overholsel Drive like over 20 because you know that there could be deer at any moment, like all the time. You can tell who drives over here regularly and who doesn't by how fast they drive down Overalls or drive at night. There's so many deer. One time, there was like a whole herd in this whole like open lot in the middle of, you know, all those houses along Overalls Drive, and there was just deer like filled the whole lot, and they're all just chilling, eating. And there was a cop that had stopped to shine like a spotlight on them and like was like pointing and like telling his partner about the deer. So many deer.
This is off topic, but it's related to microbiology. So I have a patient, well, I work in interventional GI. We deal with like, pancreatic cancers and jaundice, stuff like that. Well, I had a patient who's got referred to us for a GIST tumor.
Mhm.
And I look at his labs to make sure like his labs, and tell me why I see an aerobic. It's aerobic / anaerobic, and it says with gram stain on it. And I'm like, oh my God, we are getting this. And it was crazy, the Kirby Bauer disc and everything. But then they did like a lactic acid one.
Mhm.
But it was really weird. I was like, oh my God, like I've never seen this until now. I don't know,
Yeah.
you did microbiology.
Now you're gonna see it everywhere. It's one of those things that probably you just glance over and you don't even think about. Now you know what it is, and you're like, I know
Exactly.
why this is. I get it now.
It had all the little like what all was susceptible for like the antibiotics or whatever. But it was
Yeah.
crazy. It was I like a wound, but then it said like the Kirby Bauer disc was intermediate, and I was like, that's different. But it had like a MIC versus MG versus ML. I was like, that's crazy.
Mhm.
Look,
Yeah, there's a whole bunch of stuff that can do with it besides what we learned. It's kind of intense. All right, we'll probably get going. Mhm. Just wait for these guys if they want to join in.
Right there. Maybe I need to put it in the front. And go there.
Hmm. Why is this blinking over here? All right, guys, we're gonna go ahead and get started. Okay, I'm gonna sit down. Yeah, let's go ahead and get started. We'll play through this. Just like the other ones, and I'll give you a little info if I can remember. If I remember, but like I'll probably remember too much, honestly. I'll give you my little tidbits of info on stuff as we go, like I usually do. Probably take up too much time, but hey, more info is better than less. And I feel like you can always come back and rewatch it. And I'll give the link to the Kahoot as well, so you can play through it or play along with the video later or whatever it is. But this is the time that, I knew we had. And so we're going to do this now. And let's kick it off, whatever. Actually, I'm like, where's the start button? I'm literally looking for it. I know it's over here. Oh, it says, don't worry about saying duplicate. That's just because this was one that I edited for our purpose. Okay, all disease-causing agents are made of cells. False. But all the disease-causing agents we've learned about, do you think that's true or do you think that's false? All right, so this one is false. This is just because we know we have viruses. They are acellular, right? So they are not true cells. They don't have the right components to be cells, and they can't live outside of the cell. They cannot ever duplicate whatsoever outside of a cell without cheating and using our machinery, right? They don't have the means to, they don't have the components to. There's one other one that isn't a cell that we get concerned with anyways, and it is the prions. And remember, they're just proteins. All right, what is the correct term for disease-causing microorganisms? You guys probably know this one. All right, that's correct. It's gonna be pathogen. This is a term that we use pretty regularly. So this is probably no surprise. Just reminding you that that is in fact what the term means. The correct way to refer to the cause of the plague is Yersinia pestis. You may have gotten it for your lab report organism. So what system dictates how to write it? And this is something that some people got points off of on their lab reports. So I know we couldn't italicize, and I had mentioned that in lab, but we should still have done other rules for this one. Right, that's going to be binomial nomenclature. Capitalize the first one, lowercase that second word. Remember, the first is the genus, and the second is the specific epithet or species. And then you're supposed to italicize it. I realized that we couldn't do that on that form, but yeah. All right. Anyways. Yeah, I don't know nomenclature. Still should follow them rules. Okay, in taxonomy, kingdom is larger or more general than domain. Which one's the bigger one? All right, so that's false. Good job. So we know domain is the biggest one, and then kingdom, phylum, class, order, family, genus, and species. I have to look off into the distance while I say it so it comes out of my mouth correctly. And of course, if you need to write down like the letters in order, there's going to be one of those. Probably, I think there's one of those questions on the exam. I can't remember if I'm remembering that from the correct exam. All right, inoculate, incubate, inspect, identify. There's a missing I from the 5 I's. Which one is the correct one? I think given our choices, we should see one of them as correct, given what we know, what we do in the lab, and we're trying to work with an organism. I hope you guys like ducks. My dogs don't. Isolate is correct. So our main way that we learn in lab on how to isolate would be the quad streak, the streak plate, right? We've seen it on 2 different exams now. So. What type of media allows only certain kinds of microbes to grow and inhibits the growth of others? It's probably one we're very familiar with. No. So some of them can grow, some of them can't. What's the word for that? So that was an image of EMB. It had a letter back on color because we had such great growth on that particular plate, but they had that sheen, that green sheen on it. But anyways, that's what EMB looks like. When you get the green sheen. Yep, selective, good job. It selects for some to live. Remember, differentials just going to turn different colors to tell the difference, right? The ocular lens of the microscope generates the real image. Is that true or false? The ocular lens. This one's tricky for some people. All right, so this is false. Great job. So we know by now that the objective lens is the one that creates the real image, and the ocular lens is going to be the virtual image, because we've already gone through 2 magnifiers and some mirrors. So it's no longer, it's a big magnified version. It's not the real image anymore. What is the purpose of oil immersion? Getting through the easy questions here. Yeah. So this is going to reduce the light scatter, so refraction here. It creates a clearer image. I mean, these questions we've gone over so many times in the lab at this point. A negative stain stains the background because it is repelled by the cell surface. Is that true or false? Negative stain. All right, so this one's going to be true. The negative charge on the negative stain, the stain itself has a negative charge, is repelled by the negative charge on the phosphate groups, on the phospholipids, and other negative charge molecules on the outside of cells. So it stains the background instead of the cells themselves. So what type of chemical bond involves transferring the electron from one atom to the other? All right, so good job. This is going to be the ionic bond. Remember, we really only talked about or reprised over two types of bonds that have actual electron interaction between two atoms. That's the covalent bond where we're sharing electrons back and forth, and the ionic bond where one electron is donated from one atom to the other. Think of like sodium chloride. The sodium has the positive charge because it lost an electron, which is had a negative charge. So that leaves it with a positive. And it gave the electron to chlorine, which now has a negative charge because it has an extra electron. So that's the ionic bond between sodium and chloride. And then when you put it in water and they dissociate, that is ionization. We did talk about hydrogen bonds, but remember that in hydrogen bonds, that's more of an interaction between polarities. There's not actual electron, migration between two atoms. It's just attractions. So they're weaker bonds, but probably one of the most important bonds that we can understand when learning about, the macromolecules. All right, so carbon dioxide is an organic molecule. Is that true or false? There's a reason for why this answer is the answer. All right, so this one is false. This is just because we don't have associated hydrogen in the carbon backbone, basically. So you'll have a carbon-hydrogen backbone in organic molecules. Even though the carbon is the more important molecule, the hydrogens have to be there to support it, to make it, have the properties of organic molecules. So olive oil is a great example of an everyday hydrophilic molecule. Is that true or false? So this one is false. So when we look at how oil and water don't mix, that's because oil is going to be hydrophobic, water fearing. So it doesn't mix well. So it's probably one of those things. They're not miscible, right? That's a word they would use in chemistry. This is when we have to be sure that we're like making sure we read all the words and not skipping through it too much. Hydrophilic, man, water loving, right? Like Philadelphia, city of brotherly love. It wasn't that way when I lived there, but that's fine. Which pH would have the highest hydrogen concentration? This is about understanding what pH is really about. We'll come back to that. We can measure pH all we want, but do we understand how it works? So this answer is the green one. That's going to be 2. That's because the lower the pH, the more acidic, and acid is basically being represented by high hydrogen ions. Acid is high hydrogen ions, base is low hydrogen ions. Because pH is a negative logarithmic expression, so it's based on tens. So 10 to the 1, right, versus 10 to the 2. You might say, well, 10 to the 1 is less than 10 to the 2. But it is a negative log, so it's going to be 10 to the negative 1 versus 10 to the negative 2. So now we're going backwards on that scale from 0, right? So we're getting smaller and smaller with the bigger and bigger numbers. That correlates with what we see with the pH itself, which refers to that 10 to the negative 1 would be a pH of 1. 10 to the negative 2 would be a pH of 2. So if you get 10 to the negative 14, which is a way smaller number, away from back from that 0, that way smaller number, 10 to the negative 14, that negative 14 translates into a pH of 14, which we know is the most basic on the scale. So that's why pH works the way that it does. The lower the number of the actual pH, the higher the hydrogen concentration because of that. Yeah, unfortunately, lecture is where we ask you to actually understand that as opposed to just measure pH and tell me what's acid or base. Which level of protein structure features alpha helices and beta sheets? Okay, so this is going to be the secondary level. Primary is going to be just the amino acid sequence, just whether it's alanine, or leucine, or glutamate, or whatever it is. Each amino acid linked together. That's the primary. That's all that is. How those amino acids react to the ones right next to them in that line, that's going to form the secondary structures, those alpha helices and the beta sheets. Once those are kind of formed and we've got those structures kind of made, The tertiary structure is whenever we start having those major structures organizing with one another, like across the way, and how they're interacting with those R groups, the things that make it leucine versus phenylalanine or whatever it is, those groups shooting off of those amino acids. Some of them have charges or don't, or are hydrophobic or hydrophilic. Now those bigger groups are associating across from one another, making covalent cysteine bonds, sulfur bonds, disulfide bonds, and things like that, based on the larger structure of how those smaller alpha helices and beta sheets are going to interact. Quaternary just means that we have interaction between like one big old clump that we made from our tertiary might come to another big old clump made from another tertiary, and they have to come together to make the functional enzyme. Those parts coming together to make the functional enzyme, including any coenzymes or cofactors or anything like that. All of that makes the functional enzyme, and that's the quaternary structure. For some enzymes, very few, but some of them, all they need is what their folding is for their tertiary structure, and they can function. So in that case, technically, their tertiary is the same as their quaternary, but we really don't see that very often. It's uncommon. You usually have to see them binding with other things. All right, true or false, bacteria and archaea are always single cell. It's as simple as it feels. All right, excellent. So these are in fact single cell organisms. That's because multicellular organisms, their cells depend on being associated with one another for the organism to survive. Can bacteria form, relationships with cells like them or different from them? Of course they can. We've talked about biofilms, right? We can have them associate and even create dependencies on these biofilms. But ultimately, if you take the one cell out of the biofilm, can it live without the other? Yes. Can you take a liver cell and literally just plop it on the table, and can it live by itself even if we give it food or water? No, it has to have association with the rest of the body. It needs the nutrients from the blood and the other cells interacting with it and everything like that. So nervous system, all of that. It doesn't exist as a separate organism, right? So what cellular arrangement is featured below? Or say below, yeah, there it is. And another one pretty much straight out of lab, right? You have chains, they might be branching off of each other, but they're primarily chains, and that is strepto. All bacteria are capable of making endospores if nutrients are scarce. Is that true or false? Which bacteria can make endospores? Is it all of them? Or maybe specific ones? Yeah, so this is false. This is Clostridium and Bacillus. So yeah, just those two. All right, which group would Neisseria meningitidis, the gram stain's gonna be pictured, fall into? So it's got a nice pink picture. This is not purple. This is a gram negative. So see if we can remember our terms. Okay, so this is the gracillicutes, and now we'll break it down one more time pretty quickly. Remember I say I have to go through this in my mind, or else did I forget? Like it'll be blah, blah, blah coming out of my mouth. So Mendoza cutes. I always start there because Joey Mendoza, I think it was like 6th grade that he was my boyfriend or something like that. And he was very cute. So Mendoza was cute. And that was a long time ago. So that's archaic. So that is archaea. Mendoza cutes. Then I have the firmicutes. Those are going to have a firm cell wall. So these are all based on cell envelopes. So the firm cell wall from that thick peptidoglycan for the gram positives, they fall into the firmicutes. The gracillicutes are the next G for gram. Gracillicutes are the other one because we already put firmicutes for positive. So gram negative will fall into that one. And so we have archaea, we have gram positive, we have gram negative, which was our answer. And then we have this one left over, which is the tenericutes. And remember, tenericutes is going to represent the ones that don't have a true cell wall, like the mycoplasmas that are pleomorphic as a default. Their shape is random because they don't have cell walls to help maintain their structure. They actually have cholesterol, like we do in our cell membranes, to maintain their shape, and it's just not as effective. All right, true or false, some eukaryotes can use cilia for locomotion or motility. So for the eukaryotes now. All right, so this one is true. They can use flagella, just like the bacteria do, but they're different kind of flagella, right? They can move in a true whipping motion for the eukaryotes. They can whip. They have a membrane that surrounds the whole thing. We've got that 9+2 orientation of the microtubules in there. All of that fun stuff for the eukaryotes. That's their flagella. They have cilia, which are like oar-like structures that can kind of beat in, water-like environments, usually it's fluid environments, to help, guide food into their mouth areas, but also in order to catch them, to bring them in, but also can be used for locomotion. And then the last one that the eukaryotic cells themselves can use to move around is going to be pseudopodia, and that's like fake feet, right? Pseudo, fake, podia, feet, like a podiatrist, right? So the pseudopods of fake feet is exactly what you think of when you think of amoebas, like stretching out like a whole glob of the side of their cell and like pulling themselves along with it. That is literally a pseudopod. So they can also use those for feeding. So they could come up and pseudopod around something and then use phagocytosis to engulf whatever large particle they're trying to eat up. So they can do all of that at once. That's what we see with amoebas. Which are single cell organisms in the eukaryote family. Don't forget about, the flagella for the bacteria though. Okay, so for eukaryotes, what is the function of the mitochondria? Okay, so this is a pretty easy one. Most people know that mitochondria is the site of the energy production. It's where all of that aerobic respiration, that electron transport chain, all of that's going to be happening in the mitochondria on the folds and the cristae inside of the mitochondria. So, so important for that. Back to the flagella for the bacteria, because I just didn't want to leave it out because it occurred to me. But be sure you remember the different arrangements of the flagella around the bacteria, that sort of stuff. And remember that they don't whip on the bacteria. Their flagella are only going to rotate, and they can rotate clockwise or counterclockwise. And when they switch between, determines if they're going to run, if they're using just one motion, or tumble, which is when they switch to the other direction. And then those combinations of runs and tumbles that seem random to us, they can use that to control the direction of their movement. It's very imprecise. Whereas the eukaryotes, with their whipping motion that they can control, their movements are much more precise and direct. So, leave that out for understanding's sake. All right, so what is the term for the overall structure of an entire fungal mass? So if you're looking at the whole fungus, like this picture, that big old greenish fungi thing, what would you call that? Okay, so this is the mycelium. That's just the term for it that we got to remember. For the sporangiophore and the conidiophore, those are asexual spore-forming structures for fungi. Fungi are kind of complicated because they've got asexual and they've got sexual. And we did not talk much about the sexual reproduction of the fungi. I mean, thankfully for you guys, but they're asexual spore-forming structures. They're the ones that look like the dandelions and the paintbrush, like we saw with aspergillus and penicillium in our microscopes. Those ones didn't have coverings over them. You might say, well, like maybe they did, maybe I just didn't see it. No, you would see it. Like it would be a defined line around the whole outside of the structure. The ones without the lines, the ones that we saw in lab, like penicillium and aspergillus, those ones were kind of open and free. Those are conidiophores. Sporangiophores are the ones that have the covering over them. So again, just remembering which term was which here, covering versus not covering. And we have the hyphae, which is like when you have like the hair-like cells that are connecting to one another and then branching off and creating that characteristic structure that we think of like the hair-like formations of fungi, make it look fuzzy. Those individual branching off structures are called hyphae. That's the big picture one, that's the mycelium. All right, what is the term for the free-living, metabolically active form of a protozoa? Yeah, so we're a little bit divided here between trophozoite and vegetative. That's actually not a bad thing. At least we're thinking in the right direction here. Remember, vegetative is a term reserved for the bacteria when we're talking about endospores versus vegetative cells. The endospores are dormant and hiding out, waiting. And then we have the vegetative, that would be the active cells that are multiplying for the bacteria. For protozoa, they get their own terms. They're pretty much, for our purposes, the same concept. Trophozoite, tropho, the eating active free living form for the protozoa, and then cyst is their like dormant hunker down stage. Usually the cysts are the ones that are going to be infectious because the ones that are flowing through the water that you're about to drink that's infected, it's usually not the trophozoites, it's usually the cysts that could be in your water or whatever it is, and have some protozoa like Trichomonas, right, that causes trichomoniasis, sexually transmitted disease, that I tell you all about in Southwest Integris, how we would get a lot of hookers with that. I know I say it a lot, but it helps people remember it. So, but those guys don't really have a cyst form. They really only ever exist in their trophozoites. They're always going to be in that active state, typically. So, but remember the difference between the bacterial and the protozoa terms there. All right, true or false? All viruses have capsids. Is that true or false? Do they all have the capsids, or? All right, so this one is actually true. Remember that a virus only at the bare minimum has to have a nucleic acid genome of some kind. And there's all different kinds it could have, right? DNA or RNA. It could be single-stranded or double-stranded, either of those. And it can be positive sense, like you could read it straight off the code, or negative sense, you have to make the inverse copy and then read that. That's what negative sense means. So it could be all those. That's like 6 options right there. So those are the possible genomes we can have with a virus, but you can't just have genomes floating around, right? So those genomes, because they get degraded pretty quickly in the environment, so the genomes have to be protected by a protein coating. And that protein coating that protects the genome is called the capsid. The capsid can be the helical capsid that wraps around the genome, or it can be the icosahedral, like what we see with all the triangles stacked around one another. Those are 2 types of capsids that we could have, but you have to have at minimum those 2 things. And only those 2 things are the bare minimum. Capsid, the protein coating to protect the nucleic acid that's inside, those 2 things. You can have another coating on the outside called the envelope in the case of viruses, which I think I mentioned just a second ago, but that envelope is stolen from the host cell when we're budding out, right? So we push out and like take it like a blanket and pinch it around us. That's the envelope. It's a lipid bilayer stolen from the host and usually studied with the spike proteins of the virus. So it can just push through and then have its little spike proteins to enter the next cell. So not all viruses have an envelope. Example would be poliovirus and parvo, either of the parvos, whether we're talking about canine or human parvo. So they're very tiny. They do not have an envelope. They're actually hardier because of that. The envelope, I guess it can get dried out and can be more affected by cleaners and things like that. So viruses with envelopes like COVID and flu, they don't survive as well outside the body as the ones without, like polio and stuff, which kind of make you a little nervous when you think about it. But when you think about parvo, you ever heard about parvo and dogs that can have parvo? That the virus is actually pretty, you have to be very thorough with cleaning in order to get rid of it. If you have like a puppy come into your adoption center, like I did when I used to volunteer for the Humane Society, and everybody would bleach everything in there if it just even walked in. So, all right, the lytic phase is the phase where bacteriophages actively multiply until they burst out of the cell. Is that true or false? We've seen this one before. I know because I've seen the image. It's one of my favorite. This one's true, so it's exactly like what Star Wars was just showing us. Basically, the bacteriophage comes and lands on a bacteria, always. It does never enter. The whole bacteriophage never enters the cell. It just kind of squats down and injects its DNA into the bacterial cell. Then the bacteria, just doing its random normal existence, will start, processing that DNA and making new bacteriophages like we do for our viruses. So new bacteriophages will assemble within the actual bacterial cell, that host cell itself, until there's so many that they just burst out. They never exocytose. They never exocytose, which means like the cell would get rid of them, and they never bud out. Never. Bacteriophages always will burst. And when they're going through that phase of being replicated and then just bursting out, that is the lytic phase. So you guys remember what the other phase is, right? The lysogenic phase. And that's when the bacteriophage injects its DNA into the bacterial cell and then incorporates that into its genome, right? And when it does that, then that bacteriophage DNA in the bacterial genome incorporated in there now is the prophage. We've gone into that lysogenic state. Only temperate phages, another term to be aware of, are capable of switching between the two. And sometimes the switch is determined just by cell signals. We'll let the virus know it's time to go. So what kind of genome do prions have? Dogs have not come in here one time. Because they don't love their mom. All right, we have none, right? So this is a protein that is just misfolded. There's no genome whatsoever associated with this. In fact, you might even say, oh, well, if it's a misfolded protein, then didn't it come from a, mutated gene? No, it didn't. The gene was normal, just like it is for the normal protein. It's just having to do with the structure. So if it was like the normal protein is like this, and the bad protein is like this, and it comes near the other one because of hydrophobic and hydrophilic interactions, causes it to misfold. And then these ones go on and interact with other ones, then causing them to misfold. And so that's how it propagates. It's just by misfolded and then near other normal ones, causing them to misfold. There's no DNA issues here. And there can be certain, I could say mutations, but certain sequences in the DNA that can make you more predisposed to having this happen, but it's not a guarantee. It's not like you have this mutation and you're going to get this thing. Right. And sort of like cancer too, right? We have genetic testing for cancer. Well, just because you have the gene, it doesn't necessarily mean you're going to get the cancer. There's other environmental aspects going on with that. Similar concept, really. All right. Right. So these guys, they have no DNA and they have no RNA. They don't have a cell, right? A prion themselves, they don't have a cell, but they are literally just one protein. So if you think of like a virus, right? So if you have an actual virus, you have a whole genome coded in all these little tiny proteins called capsomeres that come together to make the capsid, right? That's what they're called. Their little subunits are called capsomeres that make the capsid. And that's all you need for a virus, but you have to have those two things. With a prion, you need just one protein, which is the equivalent of just one of those capsomeres. So it's not even a whole capsid, even really. It's just one of those units. So that could be a prion, just one single protein, misfolded. That's a prion. So there's no need for any genetic information, no structure of proteins that create a function even. No, it's just a misfolded single protein. So yeah, kind of terrifying to think about. It makes you wonder if there's other things out there that like could be affecting your health right now, or people that you knew could be affecting their health, that could have been affected just from crap like this, like misfolded proteins. There probably is quite a lot more than we understand. It's just we see the effects of these misfolded proteins. If you have a protein, that normal function protein, we know in our cell is pretty much a lot of water-based environment. So you have to be hydrophilic on the outside, and you can keep your hydrophobic crap on the inside. So hydrophilic on the outside, interacting with the water environment, and hydrophobic kind of cuddled, inside, huddled inside, safe from the water, and they don't have to see it. These prions don't do that. They will just, do whatever they like the best, and they might have hydrophobic all over their outside. That does not interact well with the outside of the cell or other cells nearby or other proteins nearby that are used to that environment the way that they should be, right? So misfold has a lot of that hydrophobic aspect, and hydrophobic like wants to bind with like. So hydrophobic, it comes up all hydrophobic to the normal hydrophilic outside of the normal protein. And then because it's so hydrophobic and likes to be that way so much, the other one will just have to accommodate it and show it's hydrophobic to it. And then that's why that misfolding. That's a really basic breakdown of it. That's the concept of it. So like wants to be near like. And so they're propagating that hydrophobic, water fearing, and then they go near other proteins, the same kind of protein, to be clear, and then cause those same proteins to do the same thing, hydrophobic, where they should be hydrophilic on the outside. Anyways, so then they bind together because hydrophobic wants to be near hydrophobic, and they create these plaques or these buildups of these proteins within the brain. And as that happens, that will cause holes to form in the brain, like Swiss cheese or like a sponge. And that's why we call it a spongy form, encephalopathy. So encephalo means brain, pathy means disease of the brain. So it's really a long word that just says it's a sponge-like brain disease. That's all that word means, spongy form encephalopathy. But that's how it happens. So these proteins would rather they call aggregate together, right? Form these aggregations and cause damage to the tissue as a result. This can happen naturally in the body just from aging, something very, very similar to this anyways. Amyloidosis or amyloid plaques can build up in your brain and cause damage to the brain and then lead to symptoms of dementia and other things. This is called Alzheimer's disease. That's how Alzheimer's functions. There's other factors involved in Alzheimer's, but it is in fact very similar to prions. It's not a true prion because it has more to do with the aging process, but very similar concept to that. There's a lot for prions. Talk about it much. We just talk about mad cow disease, but and chronic wasting disease in deer is one of the prion diseases. This is a prion disease of deer, but it's actually becoming pretty serious across the United States. And you're supposed to have, when you're deer hunting, you're supposed to have your deer tested for it so they can keep track of the spread of the disease. Okay, so Jenny has brown hair, but she has one brown gene from mom and a blonde gene from dad. But her brown hair that she actually has, what is that? What do we call that? All right, so that is a phenotype. It's the expressed one. You guys did pretty good with that one. So I'm just going to the difference between phenotype and genotype. All right, which one term describes DNA replication the best of these choices? So I'll reiterate, DNA replication. All right, so our answer here is semi-conservative. The other popular choice always tends to be transcription. So we'll just remind you of some of these processes, right? DNA, whenever you scrape your knee or you're just growing up as a human being or whatever it is, and you have to make copies of your cells, right? And they have to be the exact same genetic information. So we just make copy of the genetic information in new daughter cells. And that's called mitosis. Anytime you're going through mitosis to make new exact replica daughter cells of your somatic body cells, whenever that's happening, we go through DNA replication. That's just replicating the DNA, making an exact copy of the DNA. Okay. What is DNA for, though? Right. So we say it has your genes. What in the heck are genes? What does that mean? A gene is going to code a protein, or it can control other genes. And that's the only functions that genes have. So you make protein or you control things. That's it. Your genes do not code for anything else. Every single gene, all 20,000 genes in your entire body, either control the other genes, which is just a few of them. But almost all of the rest of them code for proteins. And the ones, like I said, that don't are just control genes. They're not gonna code for anything else. Wow, so protein becomes all of a sudden extremely important in your life. So that's all that your genes do. That is who you are, everything that you are, all of your genes encoding for who you are and what you are and everything like that, and building your body anyways. That's all just proteins coded for by your DNA and your genes. So making copies of that, making new daughter cells of that, that would be DNA replication, but that doesn't do anything for you. So if you need to make the products of your genes, like the proteins that are going to lead to you having blue eyes, like the proteins that are going to lead to you having straight hair versus curly hair, or whatever it is, right? All of those are going to be, you're going to need to make the protein product of the genes. So how do we do that? We take our DNA and we make a working copy from it of just that gene that you're working with. So the gene for having blue eyes, let's say. And you're going to make that gene copy of just that gene, and that is the messenger RNA. So that is transcription, going from DNA to that messenger molecule, the messenger RNA, mRNA, that is transcription. Then you can take that messenger RNA out of the Smithsonian Institution, your nucleus, and go out to the rough endoplasmic reticulum, studded with all the ribosomes. And the ribosomes are going to basically translate the messenger RNA message of that gene into the actual protein for the blue eyes. That protein being made up of course, amino acids. So that's the rundown of what all is going on there. We have DNA replication to make DNA copies when we need new cells. We have DNA to RNA transcription to make our working copy when we're using our genes. And then we have the messenger RNA to the protein to make our actual functional product in translation. That's done by the ribosome. And don't forget the function of the tRNA, right, which I'm sure is going to be questions here in a minute. That's going to match up with your mRNA code, basically, to bring in the correct amino acids as we're doing all of this. So that's the, man, I mean, there's probably a lot more questions about that coming up here in just a second, but we did the rundown. So building blocks of DNA and RNA are known as nucleotides and consist of a phosphate, a pentose, and a nitrogenous base. Is that correct? Is that all that correct? This horrifying image that you're looking at in this is actually RNA polymerase. So it's making an RNA or the DNA is a horrifying thing, but that's kind of how the protein looks and functions. So, but yes, so this is true. So we have phosphate, pentose, that can be the deoxyribose or the ribose. That's the sugar that's going to be phosphate, sugar, nitrogenous base. Just being specific here, 5-carbon sugar, ribose or deoxyribose. The nitrogenous base can be ATCG, adenine, thymine, guanine, cytosine for the DNA, or instead of T for the RNA, you will use uracil, U. I wrote that down, probably also going to be the next questions. All right, codon, ACA, codes for threonine, that's amino acid. A mutation changes that codon to AGA. Now it codes for arginine. What type of mutation would that be? All right, so this is a missense. You guys all got that one. Remember, silent is going to turn it into a stop instead of another amino acid, some sort of stop code. Fun sense is made up, and then nonsense is a stop. I was looking right at nonsense when I was saying the other thing. I read silent out of my mouth, and I realized that just now. Nonsense is the stop. Silent is remember nothing. You get the same amino acid as the result. Right, so if you went from ACA to AGA, but it was still threonine as the amino acid, what does your body care? It doesn't care at all. So you still get the same protein, right? So that's a silent mutation. And that happens because we have more than one codon coding for an amino acid typically. Nonsense means that you read it and it just doesn't make sense to you. And so you can't put an amino acid there. And so you stop. And that's why that would be a stop there. So nonsense just doesn't make any sense at all. Silent, you don't give a crap. All right, so what types of DNA sequences do restriction endonucleases tend to cut at? Lost ferret? You gotta be kidding us. I don't know if you guys have like ring, I have a ring security system. And people lose their pets all the time. That's pretty much all that freaking security system is about. Someone just found a ferret, I guess. All right, so palindrome, this is going to be basically like those, they read the same forward as backwards on the opposite strand. So, you know, CTT, AAG on the first strand, and then CTT, AAG on the opposite strand. And they basically bind back with one another if you were to bend them back. It's pretty crazy how it works. But that's how the restriction endonucleases will recognize where to cut. And remember, most of them are going to create sticky ends, which we can utilize in genetic engineering to stick the matching sequence on those ends and put genes into plasmids like we learned about for our transformation experiment. When we had that GFP, that's not natural in bacteria, right? We stuck that in there. All right, so what is the purpose of PCR? What's even the goal of it? All right, so PCR, the whole point is just to take DNA, a strand of it, and turn it into 2 strands to replicate it, right? And to double it up. So every time that you go through one cycle of PCR, the DNA strands in that will be doubling. I'm not gonna make you guys do the calculation on the lecture test, just FYI. Somatic gene therapy can be passed down to future generations. So I don't think I don't want to misspeak. If I do it again, I really don't think I am doing it again. I can't remember the exact questions on the test. I get them confused sometimes. Then it'll be as easy as the one that was on the lecture. So just maybe up to 4 rounds from 1. So you could easily draw it out if you needed to, but I would never do anything beyond that. Somatic gene therapy can be passed down to future generations. The somatic means your actual body cells. Can you pass down traits from your body cells to future generations? Absolutely not. Only can pass down genes that are in your gametes, your sex cells, right? So that would be germline gene therapy. And you can potentially do germline therapy if you are treating an embryo or early on in the fetal process, something like that. But otherwise, it's not going to get, you treat somebody for something and it doesn't get passed on to the offspring. Or what is the term for an organism that gets its carbon from an inorganic source and its energy from the sun? All right, so log lady is telling us that her log told her that the answer is photoautotroph. So photo being energy from the sun. And then if you can at least remember photoautotroph, right? You know that photo, the sun, they're not getting carbon from the sun. Is carbon coming directly from sunlight? No, the energy from the sun is. So then you would know that the carbon has to come from the autotroph part. So that would mean if you look at heterotroph, that would be a different carbon source, right? And then chemo being the other energy or the other energy source. So heterotroph, carbon, chemo, energy, or photo energy, autotroph, carbon. however you want to break it down. I like to always try to remember photoautotroph, and then I can't remember chemoheterotroph, because this chemo, where does your energy come from? It comes from chemicals. Oh, you're heterotroph. Okay, so where does your carbon come from? It comes from chemicals. Like, that doesn't help me break it down into which side is which. So that's why I usually will like think of it as photoautotroph and break it down that way, and then compare it to the other term. The two more important terms here, in case you can't figure it out from me just saying it out loud, photoautotroph versus chemoheterotroph. They're kind of the opposites of one another. As far as, do you get your energy from the sun, or do you have to steal it from other molecules? Which is what we do. We can get it from other molecules. We eat things, break them down, and get energy by shuttling it through the whole aerobic respiration BS, right? Autotroph, as far as getting your carbon compounds comes from, they're taking inorganic carbon dioxide, plants are, and turning it into like glucose, and they can use that for building molecules as well as energy and stuff like that. They're never stealing like glucose or other carbon-based molecules from their outside environment, like what we do with eating food or something. They're not doing that. They're using inorganic sources, which is crazy to think about. They are not ever stealing it from other molecules. Chemoheterotroph, your chemical breakdown, giving you your energy. Your heterotroph, you are stealing your carbon from other organic molecules, which is what we do. And you don't use inorganic, right? So that's the difference. All right. What is the term for a nutrient that must be provided in a diet because an organism cannot make it for itself? I'll overthink this one. Yeah, so this is growth factor. And like what was shown us, you can equate that to B12 or vitamin C, stuff like that, that we have to have provided to us in our diet. It's a growth factor for us. What type of solution could have a typical cell swell up and burst? This is the one that a lot of people were getting wrong. That's right, so this is hypotonic, very good. So just to explain it really quick so that I know you guys probably understand it, but in case people watching this in the future don't get why this is the answer. Hypotonic, I put your cells, which have some salts in them, by the way, into a Dasani water or RO water or DI water or whatever, just water. It's the purest of all the waters, right? And I put it in there. Only your water can cross the membrane. Osmosis. It's selectively permeable, unfortunately. So the water can cross freely across the membrane, but other things from inside your cell can't. So what it does to dilute the salt inside to try to equalize with what's going on outside is dilute its inside with water, and you'll take in water and take in water. But obviously this is a losing battle, because no matter how much water you take into the cell, you can't water down the fact that you have stuff inside and the water outside doesn't, right? That's ridiculous. But the cell doesn't know that. So it'll keep taking in water and taking in water and taking in water to its capacity, which for many cells could make the cell burst like a balloon being blown up with air, except in this case, it's water. So that is hypotonic. Low tonicity is referring to the salt concentration or sugar concentration, the solute concentration, right, for chemistry. That there's less stuff in the outside water, outside milieu, if you will, versus what's inside the cell has more stuff in that solution. So that's what that's referring to and why that happens the way that it does. Be conscious of what happens, whether the water outside is hypotonic to the cell. So like we just talked about, less stuff than inside the cell. Hypertonic, more stuff than what's inside the cell. So the cell is going to lose its water to the outside to try to water the outside down. And that's a losing battle. So it shrinks down to something called plasmolysis occurs. And that's what we would talk about with osmotic pressure happening when you pack things in salt or whatever, try to preserve it. And then isotonic means it's the same on both sides. Very much want to reiterate, just because your solution has the same concentration on the outside as the inside of the cell, you might be thinking like, oh, well, now we're happy, same stuff inside as outside. so nothing happens. No, the water is always moving back and forth because of chemistry, right? So it can't be as simple as nothing is happening. The water's always moving. It's just equal amount in and out for isotonic. All right, pathogens and microbes of our biome thrive at temperatures near or around room and body temp. So what's the term for that? Right, so the term is mesophiles. Meso means middle, and that's kind of a middle temperature for us anyways, right? So a lot hotter is thermo, and a lot colder is going to be psychro. Psychrophiles love growing in the cold. They thrive in the cold and probably will not do well in warmer temperatures. Psychrotrophs can manage better in the cold than most, but they probably prefer the mesophile temperatures, but they do better in the cold than most mesophiles. All right, what are coliforms? We're already here. All right, great job, everybody. I'm glad that we got this. I don't know if I say everybody, there's 3 of you answering, but that's okay. You did great with it. These are indicator bacteria. They themselves may not be pathogens, right? So just normal E. coli, not the pathogenic form. Normal bacteria that are found in the colon in general, that they don't always have to be gram-negative either. So if we talk about Enterococcus fecalis, so I think that was the gram, I don't remember for everybody, but I believe that was the gram-positive for Destiny and Nick. It's a gram-positive, but fecalis, entero, like it's in your poop, right? So that organism is a gram-positive that has adapted to be living in feces in the fecal environment. So if those organisms that are normal in anybody's poop, not gonna get anybody typically sick, except for maybe immunocompromised people, even especially in small amounts, right? So if they're there at all, then that could indicate that other pathogens could have made it through the filtration and treatment process. So coliforms are indicators that there's a risk that pathogens could make it through. I did a lot of testing with this in my last job. They would just send us literally just water samples from wherever, which you'd think your water would be the same as my water, and they're just coming from like friggin' the Applebee's down the road or whatever. But different people's pipes are different than other people's pipes or whatever. So we tested a lot of that. Right, so what is the term that refers to chemical reactions in the body involving breaking down molecules into smaller products? Yeah, this kind of gives it away, but. Yep, so this is catabolism. Cats destroy things, so they break things apart. So that's how I remember it in a metabolic process. So your whole of every single chemical reaction going on in your body, making and breaking bonds and all this BS is going on. That's your metabolism. And so you have making bonds and breaking bonds, generally speaking, in a specific single reaction. If you're making bonds, that's anabolism, so building. So think of anabolic steroids, people trying to build muscle, right? Catabolism, cats destroy things. That's the best way I can think of to memorize it. I don't mind a cat, I'm allergic to them, but they do destroy things. Not that my dogs don't, but that's not the choice. Dogabolism, yeah, that's not one of the choices, so. In aerobic respiration, what are the 3 stages of energy production? So there's 3 main stages. You just have to select the 3 in no particular order. Listen to my dogs. So hope they're not doing anything bad. I think. Yeah. Alrighty. So we have step number 1, glycolysis. Step number 2, Krebs. Step number 3, the ETC, electron transport chain. Remember glycolysis, we're just going from glucose to pyruvate. That's the goal. Make a little bit of ATP in the process. We also make an electron carrier in there. Those are not our major components, so we're really focusing on that pyruvate. But they are important to remember to make a little bit of ATP and you make a little bit of that electron carrier when we talk about fermentation, which we'll come back to. But glycolysis, pyruvate. Pyruvate gets converted into acetyl-CoA that goes into the Krebs cycle. So the main goal of pyruvate then going essentially in the Krebs cycle and shuttling that molecule all the way through the Krebs cycle and stealing off as many electrons as we can. Stealing off electrons onto our electron carriers. We're making quite a lot of electron carriers in this process, make a little bit of ATP. And because we're breaking bonds that involve carbon, we have our byproducts of carbon dioxide as well. So carbon dioxide is made, ATP is made, and the main point of Krebs cycle, the electron transport molecules, right? Electron carriers. Glycolysis, point is pyruvate so that we can make the molecule to shuttle through Krebs to make the electron carriers. That's the point of Krebs, electron carriers. Glycolysis, pyruvate, Krebs cycle, electron carriers. Yeah, you make other things, and I'd be aware of the things you have to make, but you don't need to know the numbers. Just if I were to ask you, is carbon dioxide made in glycolysis, you should be able to tell me no, especially because if we're going from 6 carbon glucose to 2, 3 carbon pyruvates, have you lost any carbons? No, so you're not making, so be able to think through it that way. Finally, the last step is the electron transport chain, which we know we're going to shuttle all those electrons from those electron carriers through our electron transport chain, through molecules like cytochrome C oxidase, like we learned about in lab, in case you're not following, like the oxidase does. That's one of those molecules that you're going to be shuttling your electrons through in the electron transport chain. That's actually the last one. So anyways, that while you're doing that, your negative electrons moving through, pumping positive hydrogens onto the outside of that membrane where all this is going on. That's called chemiosmosis. So chemicals like the protons, we're moving those across a membrane instead of water in this case. So that's called chemiosmosis. So we move our protons outside, we create that positive charge outside of the membrane, and that's not great as far as diffusion works, because diffusion and what physics says is that we want equal on both sides, which we already learned about with the tonicity, hypo and hypertonic stuff. Same thing here. So we don't want too many protons on the outside than on the inside. So it wants to equal out, wants to balance out. So it starts moving protons through the only way it can go in, which is through ATP synthase. And so that molecule, as you're moving a proton through, causes that protein channel that's moving through to go through some changes as you're having that positive molecule move through. And that leads to the phosphorylation of a DP adenosine diphosphate into a TP adenosine triphosphate, your high energy molecule. Keep in mind that all this is going on without expending any energy at all. So that's going to be called oxidative phosphorylation. It's not costing you any energy to make it. We're just using redox, or the exchange of electrons, basically, to power all of this. So it's oxidative phosphorylation here. So we can use all of that energy that we would have involved in that into plugging it into ATP. Remember, ATP works as an energy molecule specifically because of the triphosphate. Those 3 phosphate groups have a negative charge, and they're all repelling one another at the same time. Like south pole to south pole magnets, they're pushing away from one another. So that last phosphate is really pushing away from the other 2, and it really wants to come off. And when you break any bond, you release energy. Remember, when you break bonds, you're releasing energy. But if you want to make new bonds, if you want to build things, you are using energy. You have to. If you're building a building, you have to put a lot of energy into that, right? So think about it that way. Right, so we need all that energy stored up. So to build that, we have to use the energy from the oxidative phosphorylation going on in the electron transport chain. Anyways, and then when we release that phosphate on there later on in the cell, then they can use the energy released for whatever it's doing. That's the whole point of this. So we get all those protons moving in. through that whole thing, powering ATP being made. You had electrons moving through like we had with cytochrome C oxidase at the end. Electrons from there, negatively charged. Protons coming through, positively charged. Now we can put them back together just to make them hydrogens. And half of a water, excuse me, half of the oxygen, I'm giving away burying the lead here. Half of an oxygen molecule, O2, and just O with 2 hydrogens, that's a water molecules. That's why you breathe in oxygen to get rid of those built up electrons and protons from the electron transport chain. It's the only reason why you breathe in oxygen. So, because your cells need energy, basically. If you stop doing that, if you don't have oxygen for that, you can't keep that gradient and the H pluses on the outside. It'll just eventually be the same on the inside as the outside, and the electrons had nowhere to go either. So it just builds up and your cells stop working altogether because you need ATP. You can't make it without these gradients happening. So that's a lot, I know, but that's the whole point of all of this. Glycolysis, we're going to make pyruvate that's going to eventually have a molecule shuttle through Krebs to make electron carriers that are then going to shuttle electrons through the electron transport chain and via chemiosmosis, and then moving back in through ATP synthase, drive ATP synthesis. That's our energy. That's the idea. Okay, that was a lot. I'm sure all of that's going to be in the questions next. Maybe not as many as I think. What is the final electron acceptor in the aerobic respiration? This means that last molecule that gets rid of the last electrons. Can't let them build up, so we have to get rid of them somehow. All right, so in aerobic respiration, this is the whole reason that it's called aerobic respiration, because oxygen is your final electron acceptor, because it's going to be turning into O2 and basically accepting the electrons and the protons and forming water as your byproduct, but oxygen is going to allow all that to happen. So that big, you know, kind of band-aid saying you can go ahead and keep doing this requirement there is our oxygen. It's our final electron acceptor. What there's something else I wanted to say, but I can't remember. Right, fermentation generates just as much energy per glucose on average as does the whole of aerobic respiration. Is that true or false? Right, so this one is false. Remember, fermentation is just glycolysis. That's it. So it goes through glycolysis. Remember I told you glycolysis is just glucose to pyruvate, 2 of them. So 1 6-carbon to 2 3-carbons. You make a little bit of ATP and like an electron carrier, right? And that's it. That's all you get. It's not a whole lot. Not very much ATP. It's 2 ATP net when you're going through 1 glucose molecule. Anyway, so that's the energy that it would make. And it just repeats that with every glucose that it gets, and it never goes through the rest of it, ever. It never goes through Krebs. It never goes through electron transport chain because it doesn't have any oxygen at the end to deal with all the electron carriers that you build up with Krebs and all of the protons that will be going through the electron transport chain. It doesn't have oxygen as the final electron acceptor, so it just stops with glycolysis, making the 2 ATP. And they do it over and over and over and over, using up a lot of sugar in the process to make just like pyruvate essentially, but they make the ATP, that's cool, but they also have that electron carrier, that like little bit of it that they make. But in case you weren't paying attention, you can't just have electrons just chilling. We have to get rid of them somehow, right? That's what the oxygen is for in aerobic. No matter where you get it from, that electron has to go somewhere. So they don't use oxygen for that. What they will do is use that electron in their carrier, their NADH, and pop that into their pyruvate. And the reactions that go on with that pyruvate molecule, which was your yield from your glycolysis, for most cases, they're going to put that electron into that, and that leads to changing that pyruvate into things like lactic acid, carbon dioxide, alcohol, maybe other acids, mixed acid fermentation, or heterolactic fermentation, things like that. that doesn't sound familiar, it should. So that's fermentation. Who is at my door? Or just the, no, no. All right, based on what we've learned, which organism would be a true pathogen? About what a true pathogen even is. Somebody post it. Somebody was here and didn't post the thing. All right. True pathogen means that it always causes diseases, and you guys got this one right. Yersinia pestis is the cause of the plague. The reason it's a true pathogen. It's not gonna, it doesn't do nice things. It doesn't like help you digest things or become part of your biome. It's always a disease. That's a true pathogen. Remember Candida, that can be part of your biome. Streptococcus pneumonia can be part of your biome, actually, if you've been looking at our disease chapters. And then Staphylococcus aureus can be part of your biome. About 4% of people carry it naturally as part of their normal biome. So how would you know? But not Yersinia pestis. That is the plague. Don't do anything else. Okay, which term refers to an agent which came from outside of the cell or the body? Still trying to figure out who was that my door. Yeah, it was the mailman. Yeah, I live in Bethany, so I check my motion cameras. Okay, so this is exogenous. That means just outside, outside. And that can be a term that we apply to not just agents from outside of the body, but like anything that comes from outside of your body, right? So exogenous. All right, this could be more than one answer. Which of the following are symptoms? Oh no. Symptoms versus signs. All right, so symptoms are the things that you cannot measure. You just have to go by what the patient is telling you, subjective, right? So headaches, you don't have any way to measure that. Fatigue, you just have to go by, what the patient's telling you, kind of look like they're going through this thing. But fever, you can actually measure with a thermometer. Lymphadenopathy is a term that some people may not know, but that just means your lymph nodes are swollen. You can feel that, you can palpate that, right? Confirm that yourself. Okay, in which phase of infection are the very first signs and symptoms seen? 4 phases of an infection. They're all listed here. Which one do we see the very, very first signs and symptoms? All right. So incubation is the period where the virus or bacteria is growing in your body. It just got in. And from the period from whenever it gets into your body and grows up to a point that your body even cares is incubation. When you first start showing your very, very first earliest symptoms, I have kind of a scratchy throat. That's the prodrome. So, or the prodromal period. Okay. Then we have acute infection. That's where I have a scratchy throat, turns into full-blown fever, sore throat. Maybe you can't swallow, and you have malaise. You just feel like crap. The body feels run down. You're fatigued. It's full-blown, illness. And then convalescence. Your body's finally getting rid of the illness, and your symptoms are going away. Definitely know the difference between those 4. A fly flies out of the porta potty and lands on your food. It itself is not infected, but you might get sick. What is the fly in this scenario? This is probably not totally appropriate, but I was thinking about this when I saw that little video clip. That I was at a basketball game with my aunt, like my cousin, and her kids were doing a basketball tournament last weekend. And they're from Kansas. They live out in Kansas, liberal, and it's not close. And so when they drive out here, and for whatever reason, I always want to, be sure I go see them. And so my cousin was there, so I was surprised my aunt was there too. I haven't seen her in a while. And, we were talking, that was right whenever, the assassination attempt happened. And so we had that news pop up and, we got talking about politics. I'm not gonna get into that, but we were saying about how if we could choose any celebrity that we could vote for to be the president, and they were all like, I would vote for like all these like actors that had like played the president, like, I don't even know, like Dennis Quaid or something, right? So I would vote for Jeff Goldblum. I would vote for Jeff Goldblum. It seems like it would be a real riot. So anyways, mechanical vector, this means that they landed on the poop, they picked up the germs and they brought it to your food, but they weren't actually infected themselves. Infection for themselves would require the biological vector. So things like malaria, where the mosquitoes actually infected with that organism, that protozoan, then would be a biological vector. As we actually infected the organism, the pathogen is growing in the vector. Mechanicals just carry it. All right, who was involved in identifying the source of the cholera infection in 1800s London and containing that? All right, John Snow. Yeah, John Snow was the name of the guy. So he was the one that determined people getting sick from cholera. There was a pump in the middle of where everybody lived. He like dotted them out, like plotted out where everybody was sick, found out that there was a water pump in the middle, and basically took the handle off of that because people wouldn't listen to, don't use this water, y'all fools. They didn't listen, so he took the handle off of it so people couldn't use it, and then, go figure, people weren't getting cholera anymore. So that's John Snow. Remember, Florence Nightingale was the one, she was involved in dealing with soldiers who were coming into a hospital and everybody was getting super sick. It was something like half of the patients were like dying from illness. And she sorry. And let me finish that. That's how you know that I have ADHD. She introduced calling basically everything to do hand washing and stuff like that, that reduced infections significantly. So that was Florence Nightingale. And those were the two like early pre like epidemiology concepts that we have brought into our mindset of maintaining cleanliness and whatever. So the point source epidemic, those point source epidemics, for those of you who aren't in the chat, Destiny has asked, is that the point source epidemic? Point source epidemic would be one instance of one exposure at one time, and then these people got sick. So that would be the equivalent of somebody coming back from a trip overseas, coming to class, getting every single person in that class sick, and you could trace that back to that one person. That would be in that one exposure. That would be point source epidemic. A common source epidemic is what we see with that particular cholera outbreak, because it was all coming from that pump, that water pump, but people were getting sick over a period of time, repeatedly visiting that. So it wasn't one single exposure. You could consider that as like multiple exposure every time somebody went to that water pump over whatever amount of time it took until they figured it out. So that's a common source versus point source. One single exposure, you can absolutely pinpoint it to that one time in history. If that makes sense. Good question. All right, so the cholera outbreak in the this is the question. Whoa, okay, in the 1804, people got sick from the water pump over several weeks. Let's see if you guys are paying attention. All right, so because point source tells us that it had to be a point in time, one point in time, where one source where this thing came from, all at that one time, like one person coming back from overseas and exposing the whole class, and that was the outbreak. that would be point source. This, of course, common source, because multiple people are visiting this pump at different times over like an extended time period, even though it's one location, because it's spread out over time, it's a common source of water, over not just one point in time, right? So that's why that's the answer. Yeah, when the pandemic is going to cross continents, we'll talk about propagated epidemic. We're talking about repeated exposures causing the epidemic to keep getting spread throughout. So things like the flu. So that's, you wouldn't say that's a point source epidemic, right? Necessarily. Or you wouldn't say it's a common source because people can be spreading it all over the freaking place and for long, long periods of time, and it's not one source, right? So that would be something like a propagated epidemic. Propagated pandemic, like you have with COVID. An outbreak that exceeds the expected prevalence, but which doesn't cross continents, is known as. Doesn't cross the continents. Because it's epidemic, exactly. So if it crosses the continents, then it is pandemic. Endemic is something that exists normally at that prevalence, that level in your community, and it's not unexpected to have somebody get ill with it. A sporadic is just random outbreak. Like my example is always like rabies bites. That's not spreading around anywhere. So random outbreak. And then we have the epidemics, which are exceeding what you're expected to see in that area. You might expect to see like a few cases of flu, but if you have a whole lot of people getting the flu, that's an epidemic now. That's exceeding what you're expecting. As long as it's not crossing continents, it's an epidemic. All right, last question. What is the term for how many people an infected individual is likely to infect during the course of their illness? Yeah, R naught. This is an easy one if you can remember what R naught is about. I kind of am fond of this term. I just feel like it makes like one of those epidemiological concepts that like makes sense of a lot of things. You have COVID and you're likely to infect 5 people, as opposed to if you have, you know, the plague and you're less likely to infect other people. Your R naughts are considerably different. And it doesn't have to do with the likelihood that you're going to die from it or something, but the likelihood you're going to spread it to other people when you're sick. So I like it. It's one of those concepts I'm quite fond of. As opposed to case fatality rate, you know, per case, what is your likelihood of dying from this disease if you didn't get treatment? And then we have One Health concept, which is dealing with seeing the whole world, literally the entire world, and how we're treating it, the environment, and the health of ecosystems, and how that affects health in you know, developing countries versus developed countries and stuff like that. But yeah, the heart rate is not a thing. Mixture of terms. All right, so here is our podium. And if you guys have any questions, now would be the time. And I will get the link to this posted as well as the recording posted. And you guys have access to this, of course, through the entire time that you guys are getting prepared for the final. And you guys can always email me during any of your study time. Even if you haven't started studying for this yet, that's okay. You can come back to it anytime. And always email me. If you guys don't have any questions, you're welcome to go. I'll just stay on just for a couple minutes in case people do have questions. I'll get 5 minutes maybe, and then I'm gonna go ahead and sign off.
So there's going to be another review tomorrow, right?
Yes, we're gonna do units 3 and 4 tomorrow. So the unit 4 part will definitely be new to everybody. I know I reused some of the concepts from the last ones for this. I want it to be as repetitive and memorizing as much as possible for people, but yeah, unit 4 portion of it will be new to people, so. Same time, same place, same link. Lord Ash right now. So I'm gonna go ahead and sign off. And I will see you guys tomorrow. 10 a.m. Same Zoom link.